Okay, we're on to the last biomolecule. So we talked about carbohydrates, we talked about lipids, we talked about proteins, and all of those are part of our balanced diet. We have to understand those for nutrition and also for how our cells work. And the last category, which is not considered part of nutrition, is the nucleic acids, and that's because our body typically makes these. We don't have to eat them. It's not like essential amino acids. Okay, so for our nucleic acids, these are the DNA and RNA. So when we're talking about functions here, the most important function that they have is that these are the information in cells. So you may have heard of the central dogma, and I'm just going to mention it briefly. We'll work more with this later. And that is the way that information flows in cells, is that we have DNA, it unzips, and that's used as a template in the process of transcription to make messenger RNA. So this will be a whole new molecule based on the information from the DNA molecule. This mRNA is then used as a template in the process of translation, which is then used to make a protein. And this protein could be structural, it could be functional, etc., etc. The protein sequence is based on the mRNA sequence, which is based on the DNA sequence. And so we can go back and make as many proteins as we want from the RNA or from the DNA. Okay, so that's the information function. Okay, so DNA and RNA, we're just going to make a quick little chart here. I'm not going to bother drawing out the individual monomers, but we are going to talk about this. DNA and RNA. Okay, so the monomers, these always contain a sugar, phosphate, and a nitrogen-containing heterocyclic base. Okay, the sugar for DNA, that's what the D stands for, that's deoxyribose. And here the sugar for RNA, the R stands for ribose. So they differ in their sugar. So ribose is a 5-carbon sugar. Looks something like this. 1, 2, 3, 4, 5. HO, HO, OH, OH. So in deoxyribose, this OH will be missing. And that is enough for DNA to be double-stranded and RNA to be single-stranded. Isn't that amazing? Every time I think about that, I'm like, whoa. Okay, anyway. So our monomers are made of sugar, phosphate, and a base bonded down one side. So sugar, RNA also has a phosphate, also contains nitrogen-containing bases. The nitrogen-containing bases are different. Okay, so just like we saw in proteins, they had a backbone of the N, C alpha, C double bond O. In DNA and RNA, they have a backbone, sugar, and phosphate. Those parts don't change. The nitrogen-containing bases change. In DNA, that's A, C, G, and T. In RNA, that's A, C, G, and U. And the rules for pairing is A pairs with T and C pairs with G. I'm drawing lines here to represent the number of hydrogen bonds. So this is all done with hydrogen bonding. They're temporary interactions between the two sides, between the leading strand and the lagging strand. And so that's what these represent. There's 3 hydrogen bonds between C and G, which makes these a little bit stronger. In RNA, when we're making the RNA, the A pairs with the U. So if it's an A in the DNA strand, it's going to be a U in the RNA strand. And the C is going to pair with G, just like we saw there. So if it's a C in the DNA strand, it's going to pair with with G in the RNA strand. Okay. So that's what's going on there. And then, of course, we were talking about just a moment ago. Oops. Okay, just a moment ago that might as well write this down because it's going to make a big difference, that DNA is double-stranded and RNA is single-stranded. And so that's really the difference between the two. So this is double-stranded, and this is single-stranded in most cases. Okay. Let's briefly talk about where they're found in the cell, because that's going to be important for microbiology. DNA is in eukaryotic cells found in the nucleus. We're just going to focus on eukaryotes because you are a eukaryote, so you're important. Let's talk about you. So DNA is found in the nucleus. RNA is found in the nucleus temporarily. That's where it's made, and then also found in the cytoplasm. And also there's several types of RNA. One type is found in rRNA. So messenger RNA travels between the nucleus where it's made and the cytoplasm where it's read. rRNA is found in the ribosome. The ribosome is responsible for making proteins. And tRNA, transfer RNA, that carries the amino acids, that's typically found in the cytoplasm. Okay, so that's probably an abundance of information for you guys. And we have identified the structures and functions of our nucleic acids, our DNAs, and RNAs.